Yeah. This is an interesting topic, I would say, because when you get into, like we know a lot about protein synthesis because the answer to the question is what are the mechanisms that drive hypertrophy? It's protein synthesis. Protein synthesis has to outweigh protein degradation. If that happens, then you have an accrual of proteins. And, when we're talking about skeletal muscle, it's contractile proteins and structural proteins. We'll get into some of that. But um, we know so much about protein synthesis because it's directly in line with Watson and Crick and the discovery of DNA and the central dogma theory of biology, which is environmental signal going to DNA, which then codes for RNA, which makes proteins. Okay. So it's always signal to DNA to RNA to protein. And that was Nobel Prize winning science, and one of the most important discoveries we've ever had in the scientific community, and probably the most important discovery in biology. So, we understand the pieces of protein synthesis because it is going to be an activation of a specific part of the DNA. And then we make a copy of that segment of the DNA, which is called RNA. And then that RNA travels outside of the nucleus and goes to the ribosome. And at the ribosome, we assemble a protein. With this, we have the copying of the protein at the level of the DNA to make RNA, and then we have the RNA going to the ribosome, and the information in the ribosome is utilized to build a protein at the ribosome. So we have these two steps. We have transcription, which is the process of copying the DNA's code onto RNA, and then we have translation, which is the process of the RNA's information being utilized as a blueprint to basically build the protein. So that is mechanistically kind of like what's going on. And we know in biology with these things, you're always looking for what's the rate limiting step in a process. And the rate limiting step is in translation, it's not in transcription. Inside of translation again is the process of taking the blueprint of the RNA and actually constructing the protein at the ribosome with that. And we do that by going around the cell and gathering amino acids and then putting the amino acids together in a triplicate formation and stringing these triplicates along until you've built the protein. So the rate limiting factor is in translation, and we know that inside translation, because translation is like a million steps, it seems to be that the rate limiting factor is this step called mTOR, which is the mammalian target of rapamycin. And we believe that so you are always trying to keep mTOR active and you don't want mTOR to become inhibited. And it seems as though the thing that keeps mTOR active is when there's a specific concentration of leucine, which is an amino acid, present inside the ribosomal region of the cell. So when leucine falls below threshold levels, then it seems as though mTOR shuts off. And when mTOR shuts off, the entirety of protein synthesis seems to stop, and we no longer synthesize proteins.